Hi everyone, my name is Nicole Ross, and my research question is what environmental factors influence toxin production in Lingba majuscula? Sorry if I butchered that. First up, we have our evolutionary history. So, this group of algae belongs to cyanobacteria. It is a very integral part of the Earth's atmosphere by the production of oxygen by the process of photosynthesis. Through evolution, this species has become extremely competitive, which is why it is considered an invasive species. It has the ability to adapt and take over by the means of overcrowding and having the quickness to take life necessities before other species have the ability to. Some of those abilities are required for life processes, such as the use of photosynthesis, which is done by this by this algae with its ability to float and take over the spectrum of light from aquatic plants before they are able to take in the sunlight. It also allows for, or it takes away from seagrasses and corals not being able to create their own food, thus leading to death, thus allowing for more aquatic nutrients and space to be available for this algae. Characteristics of this algae are as follows, but are not limited to: the production of toxins, they are found in diverse habitats, the formation of visible colonies, and it is a prokaryotic algae, meaning it is multicellular. The biogeography, the native biogeography is in the Caribbean Sea, the Great Barrier Reef, and typically found in coastal, tropical, and subtropical marine and estuarine environments worldwide. It has expanded to the Mediterranean Sea, the Gulf of Mexico, Australia, Hawaii, Florida's Gulf Coast, and it is expanding more northern as water temperatures throughout the world rise. The physiology of this plant, their key aspects are the production of secondary metabolites called toxins, which serve as a defense and can be toxic to other organisms as well as humans if they ingest the organisms that have ingested the toxins. This algae lacks flagella and centrioles within the cell, though it does have a central vacuole and has a rigid cell wall constructed of cellulose, making it very strong. It has a distinct multi-layered sheath embedding trichomes with granulated cells where the adaptive features of the organism to cope with UVB stress. So they have, over time, developed and adapted to the stressors of climate change and the stronger environmental factors. Their reproductive biology, they are asexual and sexual, so they can produce through both. So fragmentation is how reproduction by this algae is done asexually, and then sexually, the reproduction includes both haploid and diploid stages. The haploid stages are also called gametophytes, and through this, gametes are produced through mitosis. Gametes fuse through fertilization, then in turn form the diploid zygote. The zygote then turns, thus beginning the diploid stage, which is called the sporophyte. And the sporophyte undergoes meiosis, which produces the haploids. And the reproduction of the species, both asexually and sexually, allows for rapid colonization and expansion in suitable environments, but they are very adaptable, so they do have a wide variety and their environment is expanding. The ecology of this algae is it is a cyanobacterium. It is typically found in warm and tropical marine environments, as it does form dense mats or filaments that can cover the large area, and they can be shading. So this does pose a threat to plants that need to utilize photosynthesis for food and to survive. excuse me, um, this algae can pose a threat to plants and organisms that require sunlight, like I stated, through their dense mats, and their ability to reproduce so quickly doesn't allow for expansion of other species to overcome this algae. This algae does have important ecological functions. The dense mats can be used in a positive manner. It can provide shelter and habitat for smaller organisms and fish. But again, they don't use photosynthesis to survive. This algae also does play an important role in nutrient cycling by aiding in the recycling and releasing of nutrients back into the surrounding environment. It has a very complex and dynamic ecology with both positive and negative impacts. The toxic compounds, are the Lingba toxins, but not all strains of this algae are toxic, and these variations in understanding how and why some are and some are not are still being studied. But these toxins can cause damage to organisms, tissue, and disrupt biological functions. It can kill them over time or very quickly. It is very harmful to the marine life, and once consumed or absorbed by that marine life, that marine life is no longer suitable for life or used in any manner. So this algae does have an economic impact as well as just the environment. My research proposal, the hypothesis is the increased nutrient level and water temperature to an extent will positively correlate with the growth and toxin production of this algae. I think at some extent the increase of nutrients and water temperature will start to have a negative impact. I don't think that this algae is just a forever algae and can just, deal with an increase. I think at some point it's going to cap off. There's going to be two main experiments, one controlled, which will be performed in a lab and to mimic increase of nutrients on their own, so not in an environmental factor. But the second experiment will be through an environmental phase, and these tests will be completed within natural bodies of water where these blooms are occurring. Their native habitat, their expanded habitat. So the impacts, of the nutrients will include but are not limited to the light intensity seawaters, pH, salinity, dissolved oxygen, iron, phosphorus, and nitrogen. And the reason I say it's not limited to is because we're going to start with the environmental experiment and see where and what different type of nutrients and elements of the sort are naturally found around this algae, and they may vary between, possibly different metals in the water runoff and how certain pollutants will impact this growth. And then from there, we will individualize the nutrients, the metals, the elements, and all that good stuff into their own experiments, which will be done in the lab. And here are my sources. Thank you.